Pretty good. Why are you talking about it? I feel like
All right. Yesterday, we ended with talking about the innate defenses that our body has. We're born with these things. Those macrophages, that inflammation response, those are things that everybody who's born has them. But as we know, when we're little kids, we get sick. We get sick a lot. And that is because there is a third line of defense that is called adaptive immunity. And we are not born with that. That is something that we get from exposure to our environment. And so today what we're going to talk about is that adaptive immunity. Adaptive immunity is going to rely on a couple of types of white blood cells called lymphocytes. And we're going to focus on what are called T cells and B cells. These are both white blood cells or lymphocytes. Macrophages are another kind of white blood cell, but they're involved in that non-specific innate response, whereas these guys are going to be responsible for making a specific response to a specific pathogen. Remember, pathogen is anything that causes a disease. So before we get too far, we need to talk about something called antigens. Antigens are substances that can cause a response from our adaptive immune system. So it'll cause a response from our B and our T cells. Usually, those are molecules that are found on the surface of the infection. So they're on the surface, the cell membrane of the virus or of the bacteria. B cells and T cells have matching or complementary shapes to those antigen receptors that are located on the virus or bacteria. So here's a couple of examples of that. B cell on the left, T cell on the right. And they have a little antigen receptor. Okay. That means that there would be there might be a, I don't know, virus of some sort. And that virus has a shape that sticks out like that. That was kind of bad. That receptor would match to. Okay, so let me try and draw that again. Make it a little better. So it would have a matching shape. Okay, they're so bad. Don't go there. They're so bad. But those are the antigens. This little guy here, that's the antigen. And that's what these T cells and B cells will develop, a receptor that matches the shape of that antigen. Does not like it when I go outside the line there. Each individual B or T cell is specialized to recognize a specific type of antigen. B cells and T cells will react to the pathogen in different ways. The next couple of slides that I'm going to show you, I'm not really going to explain them right now because it's going to be, I think, overwhelming if I do it now. I think it's better to go through the steps and then you can go back and look at those slides. They're going to be great slides for working on your project. If you got a question, it's like, what happens next? Well, these slides show what happens next. This is from an old textbook. This isn't in there anymore, but it shows what happens and it follows it around step by step. So step 1, step 2, step 3, step 4, all the way around, it'll show you exactly what happens. And that I really like this photo. I wish that it was still in your book. The one that they now have in your textbook is this one, which is also good. I don't know. I kind of like the other one better. I think probably because it's step by step. Even though this is probably more accurate because some of those steps are happening at the same time. This is not probably as important for your project, but it talks about what it's showing you here is the receptor that you find on a B cell. And the next slide here is on a T cell. But basically, you've got this area, this protein area that has a certain shape to it. Remember, proteins have a certain shape, and these receptors are the same. They have a certain shape, and that will bind to a particular antigen. Okay, that's really all you need to take away from those two slides. So this, I don't know why, but this is exactly the same slide as we saw the very first slide at the beginning of the discussion today. Again, just T cells, B cells, those are white blood cells. They're collectively called lymphocytes. But now let's talk about them in a little more detail. There are two, in adaptive immunity, there are two types of responses that occur. There's what's called humoral immune response, and there's what's called cell-mediated immune response. And the humoral is going to be taken care of by the bloodstream and the lymphatic tissue. So things in the bloodstream and the lymph will be responsible for this response. And they don't actually fight anything directly. What they do is they mark the cells that are infected, or they mark invading cells for destruction. And that's different from the cell-mediated immune response because these are the guys that go out and actually fight the fight. They're the ones that are going to destroy any infected cell, any bacterial infection particles or viral particles that you have in your body. We saw this before. Picture of my dude with lots of green, weird looking, kind of looks like a zombie there. But all that green, all those green lines represent vessels of the lymphatic tissue. So this is all the lymph system. And there are, we talked about lymph nodes. We talked about them in kind of in the jaw area. But there's also lymphatic lymph nodes in the armpits, in the groin area, and then also kind of below the lungs there. And those are the collection areas. So they will collect the lymphatic fluid as it fills up with, destroyed or damaged cells from the infection. And that's why you can feel those. You can palpitate those. It's the fancy way of saying feel. And you can find out whether or not someone has an infection based on palpitating those lymph nodes. That would be part of the humoral response, right? Not the cell-mediated, but the humoral. So let's talk how this all starts. So it has to start at some point. Sorry, I got something in my eye. It's driving me nuts. It's just like a hair somewhere. It's like yeah, I do right there. But it's like a hair. It's just like every time I blink. I think I got it. Okay, so macrophages. We talked about macrophages yesterday, and macrophages are white blood cells too. They're lymphocytes as well, but their job is non-specific. So if they find something that has, like that generalized, maybe it has a tail, it has a flagellum, they're like, oh, that's not right. And they will immediately attack it. But they will attack very different types of bacterial and viral infections. They're non-specific. Okay. After they do that, though, after they digest their pathogen, they're proud of their pathogen. They're proud of what they did. Kind of like, when that dog, your puppy, brings you your shoe, right? And there's only half a shoe there. You ever had that happen? I have. $300 shoes. This is like 15 years ago, $300 shoes. And the puppy's like so happy and they're wagging their tail. Look like what I have your bike shoe that you can no longer wear. Yeah. Okay. That's macrophages. Macrophages like to show what they did. And what they will do is they'll take a piece of the invasion or the virus or the bacteria, whatever they gobbled up, they'll take a piece of that and they will display it on the outside of their cell membrane. And when they do that, they are now called antigen presenting cells. They are presenting a piece of what they just ingested and broke apart.
What's the purpose?
That's the purpose. That's a great question, and I got an answer for you. That's going to start kind of the whole ball rolling, because once there is an antigen presenting cell, which is a macrophage, the next thing that's going to happen is, do I want to show that or do I want to show this next? Let's show this one first. I'm going to skip one slide and come back to that. Is these guys called the helper T cells. The helper T cells will recognize that there is an antigen presenting cell. And once that antigen presenting cell has been recognized by the T helper cell, then it's going to sound the alarm. It's going to say, hey guys, we got an issue here. We need to deal with this. So it will activate 3 other types of cells. It will activate B cells, and it'll activate 2 types of T cells. One's called cytotoxic T cell, and one is called a memory helper T cell. Okay, so the helper T cell is the one that's going to start the whole ball rolling, because now it's looking at this antigen presenting cell and say, oh, we have an issue here. There's something foreign that we need to find, and in order to find it and destroy it, we have to make some soldiers that are going to be able to attack it, and we've got to be able to recognize it and all that good kind of stuff. So the last slide that I just skipped over is a helper T cell, and that helper T cell is docking to that antigen presenting cell. So the pathogen came in here. This is my macrophage here in purple. And it's gobbled up. It's enveloped by phagocytosis. It's brought in this solid particle. And it's gobbled it up, and it's presented just a little piece, just a little antigen, a portion of that invader on its cell membrane. The helper T cell comes along, and it will form a matching copy to that antigen. Okay, so if I've got, I think what they have here is, if that's I'm just going to call it APC, that's my antigen presenting cell, then my helper T cell will say, okay, I'm going to form something that complements that. And that's going to be on the surface of that cell. That's rather large, but they're much smaller than that. So it's complementary to it. Okay, it's like, now I know what it looks like. I know what it looks like. I can tell my friends. I can tell my B cells, and I can tell my T cells what's going on. Yeah.
Is there a limit to the number of antigens that can,
Theoretically not. And that's a good question that we'll get back to either later today or tomorrow when we start talking about vaccines. That's a very good question. So, like I said, once it's got that, oh, I know what the shape is, I know what it is, guys, this is what we're looking for, it'll activate B cells and T cells. That's just another picture of it activating. All right, so, yes, go ahead.
So
yes.
On that picture, it just labels it as a helper T cell. Is it actually like a memory helper T cell?
It is just a helper T cell. It is not a memory. It is not a specific cell yet.
So once it like connects to it is a memory T cell?
No. The helper T cells are more generic, and their job is just to say, hey guys, look over here. There's something wrong. We need to, they sound the alarm. But there are other helper T cells that will store that shape theoretically for the rest of your life, and will come back out and sound the alarm if needed to. But they're like a reserve that's going to just be held in check for later on. That's a good question. So let's start with the humoral response. So B cells, that was the first on our list here. The T helper cells, say to the B cells, here's what it looks like. Here's the culprit. Okay. So the B cells are then going to be responsible for making antibodies. Antibodies are proteins. They are not cells. Their job is to bind to that antigen, to that shape that the T helper cell told them to make. So they will make themselves into a shape that is complementary to that antigen. Once they do that, they can then mark the cells that are infected and the particles that are doing the infection for destruction. So they'll go out and they'll look for that shape. And they'll say, oh, wait, here, I found one. And they attach to it. And they don't let go. Antibodies do not kill infected cells. They do not kill the pathogen. Instead, they mark them kind of like with an X that says, this one needs to go. This one needs to go. They're telling the rest of the immune system, this is not right, this is wrong, get rid of it for me. Okay, so they're directing somebody else. The peeps that are directing that are another kind of T cell called the cytotoxic T cells. Cytotoxic T cells go around, their entire job is to go around and look for cells that are marked with that antibody. Okay, but they do not actually destroy anything on their own. Yep. Sorry, I meant the antibodies do not destroy anything on their own.
So the
that's the
cytotoxic does destroy?
The cytotoxic does destroy it. The antibodies do not. Sorry, I did not make that clear. That's going to be part of the cell mediated. Good. Very good. One of the things that they do, however, is the antibodies. If this is a pathogen, this is a virus here. They're saying it's a virus anyway. The antibody can attach to the receptors located on the pathogen. And when they do that, now their receptors are bound up with the antibodies. Because they're bound up with the antibodies, they can't attach to a host cell. They can't attach to your body because they're blocked. Okay? They've been attached to by the antibodies, and because of that, they're not able to attach to anything else. So it neutralizes them. So even though this virus is still there and floating around in the body, it can't do any damage now because those antibodies are there. So back to your question about different types of antigens. So different COVID-19 vaccines, they're not all the same. So if you get Moderna or you get Pfizer or you get, I know there's a lot more now, but if you get a different vaccine, all those vaccines are working to give your body a different antigen. So they're taking a little bit of that code, actually, that will then produce a different type of antigen of the virus. So some of the vaccines might give you an antigen that's shaped like that, and somebody else might give you that might be Moderna, and maybe it's Pfizer gives you that.
But doesn't it have to be the same as the COVID?
Yes, it does. That's the thing. So the COVID-19 virus, if you look at, I don't know if this will be any bigger and you can see it. All bacteria and all viruses have lots and lots of antigens on them of different shapes. So it's kind of hard. Oh, that's pretty good. That's pretty big. So if you look here, yeah, that guy has, it's kind of dome shaped. And this guy over here is pointy shaped. Okay. So you can have an antibody, depending on what macrophage originally presented. Maybe it presented the dome, so you're going to have something that matches the dome, or you could have something that matches the spike. Yeah. Mary. Yeah, maybe in theory. The other thing you don't know about that's great. That's a great guess. The thing you don't know is how the virus is going to mutate. So it might mutate in a third different direction. It doesn't have either one of those shapes. So that's the problem. The thing that seems to be pretty consistent between strains is the spikes. So they'll talk about the COVID spikes. Where'd COVID go? There he is. So these are just a representation of some of the antigens. Of course, these are all the same shape, but these are the spikes, and these seem to, no matter what COVID vaccine what COVID strain you get, you still have these spikes on there. So it doesn't really matter which of the vaccines you get, because that spike is the one that they're really targeting. See, he's hard to kill. It's hard to kill. All right, let me get back out of here.